Now we're live.
We are live.
We are live. Welcome to, I'm just going to give myself the thing. Welcome to ChaiTime 2.5, so second season, number 5. In my new position in the house, I've been kicked out by my son from the corner over there. He set up his new birthday present with some kind of gaming system. So I moved over to the other side, but actually I think the lighting's a lot better. And the idea of keeping him in the same room as me so I can see how much gaming time he's actually doing and how much Fortnite he's playing. So you have to keep a close eye on him there. I've got my chai with me as usual, but in fact, I have my own gaming name. So if you see Big Mama, that's my new Soda Stream sparkling water, environmentally friendly bottles that we have now. So we've moved up from the plastic sparkling to reusable stuff. We look forward to a really lively session today. We are very privileged to have a number of great speakers from around the world. I'll let Jackie introduce her panelists today, but I can introduce Jackie. Jackie's been a cornerstone of the UK pharma and biotech industry for many years. I've seen her speak. She probably doesn't even know this. When I was, in my first years of business and entrepreneurship, and I was inspired by various talks that she gave over the years, and so I was really honored to have her here as chairperson today. She has a long, illustrious career within GSK. She's been head of the Research Council in the UK, and she doesn't stop and she keeps going, and we love that. So without further ado, I'm going to hand over to Jackie, and I'll probably come back in towards the end. So Jackie, over to you.
Okay, thanks, Sunil. That's a fabulous introduction, and I'm really pleased to have such a great panel here today in Jerome, Tina and Hannah from 3 exciting companies, PharmEnable, Oxford Drug Design and Recursion. So I'm going to ask them to spend just a couple of minutes introducing themselves and talking a little bit about what their company does. So Tina, do you want to kick off first?
Yeah, thank you, Jackie. Pleasure to be here today. My name's Tina, and I'm the president and COO of Recursion. We're located in Salt Lake City in the United States, which is maybe some of you have been skiing here. And I'm actually in my basement right now, but the background behind me is our headquarters. We're a company of over 170 people, headquartered here in Salt Lake, and we're decoding biology. And I think in the kind of the simple description of that is we think about building a modern biopharmaceutical company that is digitally enabled from the beginning. And so we have invested, we're almost 7 years old, and we have invested a lot of time and resources in a large robotics lab. We have a lot of wet lab capabilities, which would be on my left in my screen in the back, and have built a massive data set based on particularly phenomics-based approaches, images of biology. And we're using that to discover drugs. And so we have a growing pipeline of drugs in the rare and genetic diseases. 4 of those are at the clinical trial stage. They're phase 2 or pivotal clinical trial stage, with a pipeline coming behind those. And we recently announced this week our Series D fundraise, so it's a very exciting week. In addition, a very large partnership with Bayer Pharmaceuticals, where we're starting to look at the very complex disease areas of fibrosis. And so that's a multi-year contract to start tackling and finding new drugs for fibrosis.
Great, thanks Tina. Hannah, over to you.
Hi. So yeah, first I'd just like to say, yeah, great pleasure to join this panel, and especially on such a hot topic and something that's very, very relevant now. So I'm Hannah Saw. I'm the CEO and one of the co-founders of PharmEnable. And we're an early stage drug discovery company. We're using advanced medicinal chemistry and AI in order to basically build the next generation of small molecule drugs. So what we want to do is to make small molecules fashionable again. You know, they've sort of lost their sort of sexiness, and so that's what we're really all about. And so we're, chemical space is astronomical. It's a very huge area. And so we're mapping out chemical space and really trying to then populate that with complex 3-dimensional small molecules. And this is to really address some of the more challenging targets out there, where you do really need some new novel chemistry in order to address those. So we recently in May, again in the pandemic, closed our seed round. So we're a lot younger company for 1.8 million. And that's to start basically working on some more partnerships where we can have ownership of the assets we're now working on and building. So yeah, excited to be part of this discussion.
Right, great, thanks Hannah. And lastly to Jerome, who is the heavy duty cheminformatician amongst us, is going to be giving a slightly different perspective. Okay, Jerome.
Thanks, Jackie, and I'm really pleased to be on this panel today. It's a great opportunity to chat with you guys. So yeah, I guess I'm one of the more technical person on this panel, which I feel a little bit of pressure about that, but yeah. So I'm the cheminformatician at Oxford Drug Design. So we're a small biotech that was spun out of Oxford nearly 20 years ago now, and we've got a strong history of developing computer-aided drug design methods, initially more traditional ones based on using shape and charge to find as a similarity measure for molecules. And more recently, we've been trying to combine those more traditional methods with modern machine learning and AI techniques. And we apply these methods to our pipeline of antibiotic projects. So as a cheminformatician, I am the one in charge of maintaining our database of compounds and trying to transform that database into knowledge that we can use to inform and prioritize our resources. So yeah.
Great, thanks Jerome. Well, the whole aim of the ChaiTime is to be very interactive. I've got a few questions for the panel, but I'd like the participants to really use the chat function to chat amongst themselves, and use the Q&A function to ask questions. If you have problems using the Q&A, then by all means flag questions on the chat, and I'll try and catch up and make sure that I pick them up. I mean, all of you believe that AI is going to have an effect on drug discovery and development. Do you think we've moved beyond the hype? Do you really think now we've got very concrete examples where we can actually demonstrate clear effects on time to market, money saved, etc., or even greater chances of success? I mean, Tina, you've obviously invested a lot in the last 7 years in Recursion. What's your perspective on that?
Yeah, I think we're at least moving the conversation beyond the word hype. I mean, even the word hype, I have a little bit of a negative reaction to. I think, you know, when I joined Recursion, which was just 2 years ago, you know, and which was 4 years into the journey, there was still, you know, I come from the biopharm industry. I didn't get my background, but I was 20 years at Genentech, and so I very much am kind of the biopharma vet coming into the company. And I was certainly skeptical, but to me it was obvious something has to change, right? Drug discovery, it's too expensive, it takes too long, it's way too high of a failure rate, and then what's going to change that? And it seems obvious that the convergence of biotech and tech is the future. I mean, it feels very obvious to me, but I think the industry has been a little bit slow to fully see a different way of doing things. And so we heard a lot of this term of, you know, it's kind of tech companies hyping up the future. I'm seeing a huge movement towards the next set of questions, which is, how is tech going to change the future of drug discovery? And I think that is exactly the questions we should be asking. Our experience at Recursion, I think you can even see that even if you look at the fundraising rounds, our A and B rounds led by very forward-thinking, long-term thinking tech venture capital investors, our Series C led by a very long-term thinking investor that invested in both tech and biotech, and then now our Series D being anchored by a pharmaceutical company. And I think that almost is like the progression of going from, is this kind of tech hype, to really seeing the results coming out in terms of, you know, drugs actually making it into clinical trials. And that's where the belief starts to kind of be supported by evidence.
Jerome, Hannah, have you got anything you'd like to add to that?
Yeah, so I mean, I've lived through a number of different, what you call trendy areas and hype. So I first started in the industry, so I've got, you know, about 20 years experience in drug discovery. In the Cambridge Combinatorial, you know, the sort of combinatorial synthesis wave. And obviously that one unfortunately didn't quite live up to expectations. But then you know, moved into the sort of fragment-based drug discovery areas, and obviously that's really proved quite successful, and we've got some really nice drugs coming through. And you know, and so I think, you know, this AI, specifically AI, is sort of a bit newer, so it's going to take time. It's sort of 20 years by the time, you know, the sort of real fragment-based drug discovery, you know, drugs have actually made it onto the market. So I think, you know, I think what you'll see here is it's proof in the pudding. It's like, as the drugs start coming onto the market, and we've got things going now into clinical trials, you know, people that, you know, that have originated from AI. And I think that's the key thing. So it's about, you know, delivering on the promises. And my feeling is, you know, AI, it's a very trendy word, and sometimes it's not that it's a magic bullet that's going to solve everything. And, you know, just AI. We use AI in very specific areas, and I think that's the key thing. AI can really have a big impact on particular parts, and that's how I see it. I mean, drug discovery is always sort of looking at new technologies, and I think this is one where people have to embrace it, because it's there, it's the future. And you need to have that integrated into your companies to going forward.
I mean, you made some really good points there about the integration into the company, and I maybe come back to that later when we talk about big pharma and how, you know, as Tina alluded to, they've been quite slow to adopt this technology, because I think how you adopt it, you know, you've got some companies have gone to centers of AI, other companies have had a more integrated approach, others have kind of dipped their toe in the water and outsourced it. That's something perhaps we could come back to. Maybe Jerome, do you want to just touch on this, or we can move on to another question?
Yeah, well, I was just going to say, I mean, from my perspective, my career has been relatively short. So for me, AI seems to have always been omnipresent.
You're making me feel old, Jerome. You're making me feel old.
Sorry, not intentionally. But yeah, it's so to me as well, it seems like that it's not just the future, but it's the here and now, and it is. But we have to be very careful about the how we use it, and make sure that we combine the AI with the knowledge that chemists have, and make sure that we're not just blindly following the predictions of the AI, but actually using kind of explainable AI to make sure that we understand the predictions and make the best possible use of it that we can. To me, it's anything that can reduce the number of expensive experiments that we have to conduct, or even generate new ideas that we might not have thought of before. Anything that can do that is very useful.
Yeah, I mean, I think at Benevolent, we definitely believe that you've got to have explainable AI, and that we're really thinking more here about augmenting the drug discovery scientists rather than, you know, kind of just replacing them with a blind faith. And of course, AI is critically dependent not only on the data you have, but also the questions you ask of that data. And is that data formatted in a way that's applicable to the questions you want to ask? And this leads on to one of the questions from Simon Haworth to you guys. How much and what validation of AI-derived targets and drugs is needed before Big Pharma believes it? You know, so this sort of, I think, follows on from the discussion, because I do believe that there has been a real reluctance of large pharma to kind of embrace this technology, in part, I think, to what Hannah alluded to, because you've got, you know, this legacy of combinatorial chemistry was going to solve the world. The human genome was going to solve the world. I mean, I actually remember when SmithKline did the deal with Human Genome Sciences, there was a discussion about, first of all, we'll be able to work on all targets, and we'll know, you know, we'll be able to make any target we want to work on. And then Drake Edelson, who was then head of the combinatorial chemistry, said, you know, we'll be able to make molecules for all those targets. And I was this little voice in the audience that said, how will we know they're the right targets? And everybody kind of poo-pooed that. But anyway, great question. So we're obviously moving in the right direction. What do you think is really going to make pharma embrace this as in the same way as they embraced molecular biology? It became integral. We used to have little molecular biology departments, and it became integral of the way that pharma did things. You know, when is it going to happen that it's not going to be the specialist AI center or one AI scientist? Tina, do you want to take this one?
Yeah, happy to. We think about this a lot. I mean, ultimately, the proof is, you know, pivotal trials, right? Human efficacy trials are the ultimate proof point that we're all looking for, but that takes many, many years to get there. And so at some point in the future, I think this question becomes less and less a question because we're actually going to have the data. So to me, the question is, before you have, you know, 100 AI-derived drugs that have all gone through pivotal trials, what are the proof points along the way that kind of give us confidence along the way? And I think there's a lot of different ways to look at that, right? There's biomarkers, other things that you can look at in human, but I think even before that, one thing that's very nice, if you look at the data set that's out there of failed drugs and successful drugs, it's actually a pretty nice validation set in that sense, right? So if you have a technology and a way to think about it or validate it against drugs that have already gone into development, there's actually a pretty nice data set out there of what drugs have both succeeded and failed in humans. So I think that's another way that is like as an emerging industry in terms of, you know, tech-enabled drug discovery, we can think about not only just asking for the pharmaceutical industry to just believe, right? But are there ways that we can even think about directing, you know, some portion of our technology, not just to designing new things, but also thinking about validating to a pretty large body of decades of, you know, failed and successful drugs. I think that's one way to kind of think about it.
Yeah, the issue there, Tina, is that of course we know that there's not all the data on the failed drugs is in the public domain as to why they failed, and perhaps we can come back to that to talk about data later. That I would like to move maybe a little bit onto the chemistry side, because I have a particular interest, and there's a really good question about whether or not the patent system we have now works for AI-enabled inventions. So my question, and then perhaps you could answer the question of the participant, how much do you think this technology will allow us to open up chemical space and new synthetic methodologies? Because I was shocked when I suddenly realized that it was only like about 35 or so, plus or minus, synthetic routes that were generally used within the pharmaceutical industry. And when you talk to people like John Overington, the actual amount of chemical space we explore is quite small. So, you know, is there an opportunity for really radically redefining, or even from a small molecule perspective, and hooray for small molecules as well as big, you know, what we can actually do chemically? Jerome, do you want to take that first, and then maybe Hannah?
Yeah, well, I think it is a difficult problem, because obviously a lot of these kind of methods rely on the data that's already there. So you kind of end up, even with something that's based on synthetic methods that already exist, it's going to be difficult to extrapolate new methods from those. But I think there's still some scope for doing so, or for finding new molecules that might be synthetically accessible, but not the pathway hasn't been found yet. So I think there is some progress that can be made there. But like I say, it's if you're depending on stuff that already data that already exists, then it's going to be a challenge, but not one that I don't think can be overcome.
Hannah, do you want to?
Yeah. So I was going to come back to your point about big pharma as well. So the discussions I've sometimes had with big pharma is they don't want to be told again and again, you're doing it all wrong. So I think it's about how we sort of like interact and things like that. And for us on the small molecule side, you know, as I said, chemical space is astronomical. The vast majority is scaffolds. I can't remember the percentage. I mean, the majority of the scaffolds in Cas that are known, all the compounds are based on literally a very small number of scaffolds. So the vast majority of everything that's known is very, very limited. And I think that comes down to maybe a bit of the history back to combinatorial, where it was millions of compounds, but all on very sort of similar chemistry. But it's also about, linking that in with academia. So I come from Cambridge University. It's coming up with new methodologies and new synthesis and new routes that are robust enough that can go into lots of different types of chemistry and to create lots of different molecules. So at PharmEnable, what we focus on is taking inspiration from nature. And so nature starts off with very small things and then builds up the complexity and diversity in a small number of synthetic steps. And so that's what we really focus on. And you can, in a very sort of relatively small number of steps, come up with quite complex and diverse molecules. And so that's really where we are and what we're doing.
Could you both comment on the patentability issue? Because we've had a couple of questions on patentability in terms of, do you think that actually we've got to think about things a bit differently, or do you think that there's going to have to be is it going to become an issue really? If you sort of said that actually, how much human input do you need for novelty and invention really?
So when it comes to patentability, I think it depends what you're trying to patent. So I mean, for us, we don't try and patent the algorithms and that behind it. We still patent what is, I would say, traditional, which is the composition of matter patent. So, and that itself, we patent it when you've got the stage where you've got evidence and that around it. I think we can strengthen the patents because we can also showcase and predict the activity of very similar compounds all around it, so to strengthen that. So so I think in terms of patentability, it still comes down to the composition of matter patent, and with going into completely new chemical space, that makes that easier because it's not another benzimidazole, for example.
But even if it was a machine that decided what that chemical space was.
Yes, but you would have made the compound. So the whole point is that you have to come from the AI and the virtual world into the real world, make the compound, test it, and so it's about.
No, I mean I completely agree with you. If you want, you can comment, Jerome, or I've got quite a technical chemistry question for you, if you would like. We're just sticking with chemistry for the moment. We will move away from chemistry and give a break. Okay, so this is from Hamza. There are multiple possibilities for molecular data sets. So you've got SMILES, MOL files, SDF, etc. Would you please explain which one is suitable for extracting features? For to be able to look at them at active group, chirality, So if you and it also links to another question, which is around molecular docking. Do you think AI can improve predictions of molecular docking? So I'm fielding that one to you, Jerome.
Well, I guess it depends on the kind of things that you're trying to get out of your data. But so we use SMILES and SDF, but because the SMILES string is a 2D representation of a molecule, you have less information encoded in that. But one of the things we do at Oxford Drug Design is we have methods for generating conformational ensembles of our molecules. So converting that 2D information into a 3-dimensional representation that better captures the kind of shape of the molecule and the distribution of the features. So it depends how, but obviously that's more information rich, but requires more storage space, and so it just depends what are the important features of the molecules that you're looking at as to what representation you should use, I guess.
So it comes down again to what question you're asking, really, doesn't it? I mean, if it's activity, then you want a particular set of, you're better off using something like smiles, I suppose.
Yeah.
Okay, actually there is one question I will ask on chemistry, because I think this is a great question. How can a biotech chemist with little knowledge of AI and a restricted budget use it to impact projects? Because that's actually the problem that's facing a lot of people at the moment. What advice would you give to that biotech chemist? And feel free to chip in as well, Tina, if you've got a view.
Yeah, I mean, I guess I would say, in terms of being a biotech chemist, our industry needs a lot of people that work at this intersection of biotech, chemistry, technology. So I think as from a pure kind of talent perspective, that's a lot of what we spend our time on, thinking about building like the workforce of the future, which is chemists that understand software engineering, software engineers that understand biology, data scientists. So please lean into learning more about this area. I do think the challenge, and it's maybe an answer even to some of these other questions, is you need a lot of data to really do. I mean, whether you're talking about AI or machine learning, these tools, I think where some people, where they have potentially failed in the past, is people have tried to apply these tools with just too little data, right? And so I think that's the question that I always, and some of my former colleagues in the industry talk about, oh, we have a lot of data. It's like, do you really have a lot of data? And it's like if you're talking terabytes, you don't have a lot of data, right? Do you really have a lot of data? And I think that's the challenge of it. It's hard to just kind of dip your toe in the water unless you have a really, really rich data set to apply these tools to. So if you don't have enough data, I think don't give up on the tools, or don't give up on AI, just because maybe you get an overfit model due to poor data set.
Well, I mean, one of the ways I would think of is either collaborate with a company that does know the stuff, and they are usually flexible. There may be some milestone payments, etc. It doesn't always have to be a lot of money up front. You may even share on the downstream in terms of royalties, etc. Be creative, but also academic collaborations can help a lot to help you understand more about AI and its impact. You don't have to be a data scientist necessarily yourself. And I completely agree with Tina. These people who we call benevolent, like product owners, product managers, but really span that divide between the domain expertise and the data science, they're like gold dust. You've got to love them. Jerome, Hannah, have you got any advice for this poor lone chemist?
I would say I would want to know exactly what the question is you're asking, as to say if where AI could actually really help. And I would say reach out to people and speak, and to try and partner or collaborate, exactly as you said. So I think what's important is that AI is now being integrated, and I think that will help it being integrated through into pharma, and everything is being integrated and really talked about and learned more in universities now. So in Cambridge, there's an AI part that's now actually AI PhDs within chemistry, not siphoned away in the chem informatics part, actually in with the wet chemistry and everything. So and I think that's where it's having that crossover, and that integration is where it's really going to help. But for that lone bioinformatician, you can always contact us. We at least we can speak to you and see if there's anything we can help with.
And that's good advice. Find a mentor who knows about AI and the pharma business, and just ask if they can give you some advice. And I think that actually leads on to a more general question. I'm going to come back to target validation and data lately, but one question I don't want to lose, having you guys on the line, is you've all been involved in startups at various times. You're at various stages of the process. What's the greatest hurdle that you have had to overcome in your company And do you have any advice for new startups? Because obviously, you know, we've got you here, and it would be really good to hear your words of wisdom on that. Hannah, do you want to kick off?
Yeah, sure. I mean, starting off, I think this might be the difference also possibly with the UK and over the pond as well. But over here, I think one of the biggest challenges is funding. And in order to get funding, it's really important that you've got data and validation and things behind you. And so for us, that was one of the main things. It's how to get that on you know, with very little money. And so I think that's one of the biggest challenges. And I would say on that is you just have to use all the resources you have around you and to be as smart as you possibly can on that. So, you know, try and do some pro bono stuff with a larger company to get that validation that you can then take to the next deal and keep building up from there. Thanks.
Tina?
Yeah, so I think, I mean, funding and maybe looks easy on this side, but it certainly has been a challenge in building the company. And I think so we've obviously been really blessed to be well resourced. But I guess the tip I'd give in terms of funding is pick investors very carefully in this field. Particularly a lot of the tech VC type investors are looking for a relatively short return. Developing medicines is a very, very long game. And so one thing that Recursion has done brilliantly, and I wasn't here at the beginning, so I take no credit for this, but our CEO, Chris, was very thoughtful about cultivating even tech VCs that had very long-term horizons. Our Series C, Bailey Gifford, which is actually out of Scotland, is a very long horizon thinker, as is Bayer with a pharmaceutical company. So I think it's the type of funding and who funds you matters a lot, because you need to find those funders that are going to have the patience to see this all the way through, because it takes quite a long time. The other thing I would just mention is talent, is kind of this we mentioned before, finding people that like deeply understand biology and machine learning and software engineering and chemistry and data science and automation. Obviously nobody has all of those skills, but we actually spend a lot of time doing 101s. The data scientists do 101s for the biologists do 101s for the software engineers, et cetera, just to kind of try to create a somewhat cohesive team that actually can like understand multi-facets of the science and technology involved here. And that's a pretty big challenge to get everybody on the same page there.
I think the investors comment is really important. I think you have to pay more due diligence to your investors than you do to your life partner decisions. So, and also remember to have the prenup. So know what you will do if things go horribly wrong. It's not the discussion you want to have, but sometimes you need to have it. Jerome, any other final thoughts from you?
Well, I was just going to say that as a chemoinformatician within a small startup, my main issues are both resources for computation and data to build those models. So I'm quite lucky at ODD that we have had the foresight to have purchased some pretty decent servers that have got quite good computing power. So we have some of that in-house. So I'm quite lucky in that respect. But the data has been coming over the years, and so quite fortunate as well that our main antibiotic project's been going on for some time now, and we're at a stage where we have enough data to be able to build some usable models on that data. So it is just a question of time and resources, as the others have said. It's got to make the best of what you have.
So I've got a question here now for Tina, really. You touched on it, I think, at the beginning, that the return on investment in pharma is poor. I mean, personally, one of the reasons why I'm so passionate about AI is that I actually think the industry is kind of unsustainable longer term, because the ROI has gone from 10% to 1.8% over 10 years. And so, especially where you sit, Tina, do you think that the contribution that AI can make to better understanding of disease biology, better target validation, will have a real impact on this return on investment, i.e. are we going, not just improve the chance of, cut the cost of failure by making fewer, better compounds, but actually understanding the target better?
Yeah, so I guess I would say, yeah, the return on investment, I mean, this is my worry about our industry. You can't have, I mean, at some point, the pharma companies right now are better just kind of putting their money in the bank, essentially, right? And that's what that ROI calculation tells us over the last 10 years, than actually investing in R&D. And that is an unsustainable way for the industry to move forward. And so we have to change something, right? We cannot continue to survive with a 90% failure rate in clinical trials, not to mention all the patients that are impacted on failed drugs. So I'm very passionate about the industry needing change. And I think for me is what changes industries, right? And this is very informed by, being at Genentech in the 90s, recombinant DNA technology was one of these technologies that actually totally changed the industry. And having been at a company that was kind of leading the charge of that, there was a lot of skepticism of that technology. That it would seem like this kind of like niche technology. Was that actually going to be meaningful? And now, if you look at the top, pharmaceutical drugs in the industry, they all derive from, or most of them derive from that technology. So, disrupting the industry is very possible. But the question is, how do you disrupt the industry? And to me, what this tech-enabled drug discovery, I avoid the term AI a little bit because I think some of that hype comes from this AI. It's not just AI, it's like new technologies like CRISPR, right? We have new tools to manipulate biology. It's all the chemistry. I love, small molecules are sexy again, like this, this chemistry space we just haven't really had the tools to explore previously. Now we have these tools to explore biology in a new way, explore chemistry in a new way. And anytime you bring a whole new set of tools to bear with all of the cloud computing that's available now, the computational power behind that, you're going to see the world in a new way, right? And I think the hard part is that new way that we're going to find a whole bunch of new targets. I mean, one thing, we actually are pretty proudly target agnostic in most of our approaches. What we care about is finding drugs. And if the target's not immediately obvious, we worry about that downstream. And so I think it's just like this, it's a different mentality for how you go after drug discovery. And yeah, and I believe that will change that equation, open up a whole new set of thinking that just we haven't had access to.
So I mean, essentially, Recursion's approach is really one of using phenotypic screening, but being able to do multiple parallel analytics on the output to enable you to do multi-parameter optimization of your molecules, presumably.
Yeah, and we've definitely, our big investment's been in phenotypic screening. So we have a large image-based data set. We are investing in what we call orthogonomics, right? The various kind of omics assays that would supplement that data. And the way we just think about it, we want to create a map of human biology, right? This all starts with human cellular biology, and we do that in biological contexts with different chemistries, large molecules, co-culturing cells. What are all the ways that we can generate, create this kind of the Google Maps of biology, right? So that you, over time, get enough data that you can start inferring, even without running the experiments, you can start inferring, how to, what new scaffolds might be interesting, as an example, to kind of explore in areas of biology. I'm an engineer by training, and so to me, anytime engineers or scientists get a new tool, it opens up a whole new area of exploration, and you don't always know exactly what you're going to find, but you always find something new. So that's a little bit of my philosophy of life. So.
Yeah, I mean, I think it's amazing that you can ask questions you could never ask before, I think. And one of the other questions was around toxicology, and I think this is a rather underexplored area. To what extent do you think this technology could help us in terms of predicting off-target toxicology? Anybody want to take a hit at that one?
Yeah, I mean, that's a really relevant area, and something that a lot of companies are particularly looking at, including ours, where we, that's where I think more data here is very, very valuable. And that really feeds into understanding what the potential off-target effects are. For us, obviously, there's still a balance with when you've got very new, novel compounds. you know, the data that's out there is sometimes around the existing traditional scaffolds. So, you know, we've worked hard on trying to how to apply that to completely novel scaffolds. But that's an area, I mean, you know, getting selectivity and getting more complexity ultimately will lead, we think, to better profiles and hopefully less attrition in clinical trials.
Okay, I mean, I think there's a lot of questions coming in. We've got to think about data. To what extent do you think, we've talked about the importance of data, to what extent do you think that data, the data sets we have are fit for purpose? And I especially here would highlight negative data as being important for learning. Anyone want to kick off with that one?
Definitely for us, all data, but curated data. The more data, the better you know, it is. So, and it is. You're very much right there. It's negative data is incredibly valuable in order to really properly train AI models. And so I think this is where I always sort of, you have AI, you have where it can have a big impact is in the target identification, in the molecules, and then also in the patients and the patient populations and the patient stratification and being able to identify the ones it's likely to have a good impact on. Yeah, so that's...
I mean, at the end, I'm just giving you fair warning, panelists, that I will be asking you where you think that AI will be having the biggest impact in the short term and the near term, so you can be cogitating upon that as we go through. Do you think that we're going to see effects? I mean, there's been claimed, like Deep Genomics claimed the first AI-enabled drug in clinical trials, but it wasn't a drug. It was I mean, it is a drug, but it's basically a small molecular RNAi approach. Are we behind in applying this type of technology to antibodies, for example, or to other modalities? Obviously, I mentioned the Deep Genomics example, which is a good one, but you know, is it always going to be greatest benefit to small molecules, or is it going to be equally applicable in terms of the design? Now, clearly in things like clinical trials, patient stratification, etc., you know, that's going to be across all modalities, but in terms of creating the entities themselves.
So I'm aware of other startups that are using AI for antibody discovery, and maybe not just the whole antibody, specific regions of the antibody, so you know, bispecifics and things like that. So yeah, so I think it's, I mean, I'm not an expert on that area, but that's definitely, I can see that it's going to have an impact across the whole of drug discovery. Where I always say is it's not just machines and everyone else. It's about the integration, and that's really, really important. It's about using the data and not just blindly following the data. It's using that with very experienced and talented scientists, and that's where we're going to get the best out of it.
So that's actually a good point. Unless the others want to comment on, you know, I think it is important. It's going to be important for all these modalities. But how have you managed to really, within your organisation, get the data scientists and the domain experts working together? I mean, at Benevolent, I'd say it took at least 2 years before we moved away from the data scientists thinking they knew what the domain expert wanted, going away, coming up with something that they could do, and then presenting it. And then the domain experts didn't actually ask the data scientists for what they really wanted, because they thought the data scientists would never be able to deliver it, and so we're kind of accepting stuff that actually wasn't really the right stuff. And it was only when we got real cross-functional teams and shared goals and objectives that it really kind of took a step change, and had those translators, those product owners, bridging that gap. Is that your experience, Jerome, Tina? Has that been your experience?
I guess from my perspective, at Oxford Drug Design, we're quite a small company, so the connection between us is well, it used to be just across the across the room. So it was much easier, I guess, to have that connectivity between more chemically minded people and the modeler like myself. So, but I know of other larger companies where that is has been much more of a challenge of get it, and in academia as well, it's much more of a challenge to get to cross that divide. But yeah, I've been lucky to not have to.
So
deal
small
with that
is beautiful from that point of view.
Yeah.
Yeah.
I would just say small is beautiful. Yeah, Jackie, that really resonates with me. Particularly as our company has grown, I feel like we constantly put energy into the system to maintain those cross-functional ties. Yeah, one of our company values, we call it one Recursion, which is like that kind of constant reminder that we need to work together. And I think a good example of that is thinking about how we design and then analyze experiments. We're running now up to almost 1 million observations every week. The pipeline goes 6 days a week, and actually the 7 days kind of for troubleshooting, if you will. And so we are constantly, needing to design experiments, analyze experiments, and every one of those teams in terms of the upfront design, data scientists design the experiments up front, but obviously need a ton of input from the biologists and from the chemists, from the automation engineers, right, to make sure that the experiments are going to yield data on the back end. And similarly on the back end, when everything's come off of the platform, we do quality control. And once again, it has to be a cross-functional team looking at the quality of the data, both from a data science perspective, from a biology perspective, from an operational kind of quality control perspective. And so like creating those models where every team is thinking cross-functionally, but I feel like it's one of the things that we constantly have to be putting energy into to make sure
people
that
naturally they kind of revert back to their comfort zone. So when the tough gets going, people go into their comfort zone, and it's really getting them out of their little niches. Now
we do quite a lot of that in O2h. We have obviously we have the discovery scientist team, and we have the technology team. And when we first sort of set up the technology team, they wouldn't talk to each other. They'd have their lunch separately.
Wear their headphones.
Yeah, exactly. The headphones, and the cultural aspects of the teams are different. But Prashant mainly is my brother. He's spent a lot of time in the team, and you force them to work together, and for the chemists to understand how the tech work, the technologists to understand how they make a molecule, really work through that process. And because we're all in one sort of big building in Ahmedabad, we were able to do that quite well, I think, and very well. So we have a fairly significant sized team, but they've all been working together now in technology and chemistry, and all the meetings are joint now for the last number of years. So yeah. I think that is where where this biotech meets technology is where we could have huge impact. I completely agree with what Tina was saying around the cost of discovering and developing drugs has to change, and technology has to have that impact, whether it will be quickly or it will take some time to get there. It's great to see companies like Hannah's company and Jerome's company coming, emerging from the ashes in a way, or from the ground, and building their companies up. It's great to see Tina, with a $240 odd million of funding this week to take it to the next stage, and having a number of drugs that have come through her platform going into the clinic. And I'd like to sort of just on that note to ask you, Jackie, you've been working with Benevolent, which is and you were board of Benevolent, which is one of the probably earlier companies in the UK in this space, and one of the most significantly funded companies in the UK ecosystem, generally in biotech. So they've obviously been through, I mean, I remember a lot of the team members that I used to be part of Cambridge Biotechnology years ago. A lot of the people went through Proximogen, and somehow a lot of the people there are sort of, you know, hardened scientists and biotechnologists. But then it had this AI machine learning wrapper that sort of was brought into it. So where are you with Benevolent? Because I don't really, it'd be good to hear from you where the company is currently at and where do you think it's going to go?
Well, I mean, at the moment, I'm just going to switch so I can see myself. At the moment, as you know, we had a very successful round last year. We've got Temasek in as a, you know, like Tina says, it's important to have investors who are in there for the long term. And we've been expanding our portfolio. We don't talk publicly about all the disease areas we're working in as the portfolio has grown, but we know we have a major collaboration with AstraZeneca in IPF and chronic kidney disease, and we have a collaboration with Novartis. I'm just actually blown away. And the thing, there was a question which others can also answer. They wanted 3 examples, but I'll just give one about what sort of blown away is we published early on in the year in February. Joanna Shields, who's the current CEO of Benevolent, sort of asked one of the scientists to look into, you know, is there anything way we can use our knowledge graph to come up with some novel insights for potential therapies for COVID? And, you know, within a day, he was able to kind of pull together a really high quality selection of molecules and identify in that some molecules that were not only anti-inflammatory, but one in particular that was reasonably safe and well tolerated that also potentially could be antiviral. You know, and that's the speed of that is just amazing. And that molecule has gone into, it's an Eli Lilly molecule, baricitinib. They essentially validated a lot of our hypotheses that we came up with in terms of the in vitro biology, and was sufficient. And then we with some investigator-led studies came up with some positive data. You know, that molecule is now in clinical trials, both with recitiniv and on its own in an Eli Lilly sponsored trial.
I mean, you come from a very traditional pharma background over a number of years.
But I've always been a bit of a renegade, you know.
Yeah, and so are you a complete believer now in the AI platform story?
Oh, God, yeah. I mean, one of the, I mean, it's
because you see a lot more than we would see.
Yeah, I mean, to be perfectly honest, I think a lot of this, 50% of this is social science. It's about getting people to adopt and embrace the technology. Again, coming back to an early example when we started out, you know, when you start out doing this stuff, it doesn't always work. It is clunky, and then it's so much easier for people to revert back, you know, the medicinal chemist to go back to making the molecules or designing the molecules they always did, rather than hanging on in there and making the tech better. I fundamentally believe that this technology will cut costs because we'll make fewer molecules, we'll make better molecules, we'll know what their properties are, and we can do a lot of things in parallel with multi-parameter optimization, but also to pick the right patient group, you know, and I'm really hoping that we can be more successful at working on the right target. And because that's really the killer, but also understanding that right target might not be the right target for every patient. And then we're going to have to think about innovative commercial models to bring those sorts of things to market.
On the commercial models front, what I've sort of seen over the very short number of years that AI and machine learning have been impacting biotech, when companies first start up, they try to do collaborations with pharma, and I'll be interested to hear from you and Tina as to what those collaborations can look like, and that might help Hannah and Jerome as they build their companies. But also, has it moved, as my perception is, it's moved from doing collaborative deals with pharma to doing more in-house research and working on the fruits that come up off your platform, taking them to a much later stage and then looking to collaborate with pharma, rather than collaborating off the back of what comes off the platform?
Well, I mean, I think our original intent when what was Stratified Medical and became Benevolent was set up was to use the platform and primarily to identify molecules in the same way as Recursion has to a certain extent, to repurpose. And in some ways, I think we were a bit too early because pharma was still in the mindset of fear of success. And when I say fear of success, it's if I out-license this molecule and it works, I will get pilloried for it. So it was really hard to get molecules that we had identified, get molecules in at a reasonable upfront to enable us to be able to progress them. So that's when we shifted our emphasis. We still collaborate with pharma, but in to target ID and validation, but to also use the tech to in disease areas where you can, with a smaller sample size, be able to take a molecule through either 1B or phase 2 and show proof of concept, which I think is the model Tina's adopting. But you're also seeing it with companies like Schrödinger, who now have their own drug discovery, and Exscientia, and a number of companies see that the tech is actually the potential of it to make a difference is so great that by just partnering on a almost fee-for-service basis or whatever, you're not going to create the value that the tech can deliver. And in a way, I think the fact that companies are adopting those models or doing collaborations with downstream milestones and royalties really speaks to the fact that there's a lot of it's not trust, it's certainty, a lot less risk. There's a lot less risk with this approach that you will be successful.
Well, I see when tech investors look at our space, they're very positive with the pharma collaborations, even if they're very early, because it kind of validates the platform. But then they quickly realize actually the values, pharma companies, ultimately they want to buy drugs. They're in the business of selling drugs. And the closer you get to something that looks like a drug, there's some huge value inflection points that you go through.
Well, I was going to say, when we were talking to investors in the round before the last round, the tech investors, when you said, oh, somebody could pay 200 million up front for a phase 1 molecule or phase 2 molecule, they kind of, it was just sort of really foreign to them. Of course, now what happens is the tech investors have become much more healthcare and pharma savvy, and investors like Temasek, like SoftBank, they have huge arms that are very.
I've seen some of the more recent rounds into, Tina can probably comment on this because she's just done one, but I've seen some of the more recent rounds in what I would say is very traditional biotech drug discovery research with an AI platform being completely taken out by tech companies. And I've seen this huge wall of cash come into the sector from tech investors, Tencent and, things people you just wouldn't consider as being typical, into a biotech drug discovery play. And I think that's brought in a whole lot of new money into the sector, but they've seen that, okay, you may have to wait a bit longer, or the risk may be less understood, but the returns are certainly there. Tina, can you talk to me a little bit about your Bayer deal, first of all, and then I'll ask you a little bit more about the question on tech.
Yeah, and I'll pull on this point that Jackie made about fee for service. I think this is a very important decision that a company needs to make early on, right? Because I think, particularly if you come in tech investors, this fee for service concept resonates a little bit more like software as a service, right? That kind of understanding that you get in some revenue. But Recursion made a very clear and conscious choice that we are not a fee for service company, and we don't take fee for service collaborations. And it takes a lot of kind of funding and resilience in some ways to hold that line for many years. And so we think about ourselves as a new generation of pharmaceutical company that will collaborate with large pharma, right? And so that kind of feeds into the Bayer deal, and that being a collaboration.
How that deal was structured. It was a mixture of investment and collaborative development to it.
Yeah, so it's both. So they did lead our Series D, so they put a $50 million investment as the lead to our Series D round. In addition, the deal was structured $30 million up front, to kick off the partnership and some investments we need to make there. And then milestones. So we have over 10 programs kind of outlined with them, all within we're exclusive with them in the area of fibrotic diseases, particularly starting with lung, kidney, and heart. So over 10 programs, each of those programs have milestones of over 100 million potential. And then for anything that makes it to commercialization, royalties downstream. And so just kind of thinking about that structure and the economics of that's not a fee for service structure, right? That's two partners trying to find solutions to fibrotic disease.
Did you start off when, earlier on by trying to do CRO plus deals or small collaborative deals with the pharma rather than these more significant deals you have to do?
We got a lot of interest in those type of deals and always turned them down. And that's why I say it's, look, I'm not the CEO, I'm the COO, right? I watch the money go out the door every single day, as part of my job. And so, huge credit to Chris and the rest of the team to have that vision and that resilience to really hold a line on, we would rather develop our own drugs, right? Which is, we do have, once again, 4 repurposed drugs in the clinic and in the area of loss of function genetic disease, to get to a point where we have demonstrated our abilities that then interest these pharma companies in doing these large scale collaborations. So huge amount of credit to Chris in particular for having the patience
we are getting towards the end of our time. We're actually 15 minutes over, but Jackie was doing such a brilliant job of teasing out great information. I had to just let it run. But I would, really want to say thank you for Jackie for chairing the session. She's, it's our honor to have you on our ChaiTime.
Thank you. That's very
really
kind.
Appreciate that. I would say that, Jerome, Oxford Drug Design, Hannah has some great answers there. It's good to have both perspectives. Hannah has just completed a funding round of over $1 million, and she's now looking about how she develops her partnerships, and maybe she'll have to do some CRO plus models, I think, to establish the platform. But I think she would like to do her own drug discovery as well, and she's trying to work out how to make that happen. She will probably be doing another funding round, so if you're interested in PharmEnable, then do send me an email and we'll put you in touch. And Jerome is part of Oxford Drug Design. They have, he's been quite shy about the success that they've had. They've had over an $11 million of soft money from Innovate UK, mainly from Carbex and others, and which is entirely funding the antibiotic program towards the clinic, which is doing very well. And their platform lends it, they've actually got a drug that's come off the back of their platform. So a lot of AI companies that we see pitch, they actually have a platform that actually they've been doing this many, many years. They're very low key, brilliant scientists and technologists based in Oxford. And they're going to be doing a funding round as well to start other new programs on the back of their data sets and platform that they've been working on over 20 years, which came out of Professor Graham Richard's lab at Oxford. So if anyone's interested in investing into either of those companies, then do drop me an email or someone in the team. I'm sure you know how to get hold of me through LinkedIn and other ways. Tina, huge congratulations. You must be inundated this week. It was a humongous deal that we could have picked a better time to bring you onto the ChaiTime. The deal announced was like 200 something. That's a colossal deal. It's taken, it's one of the biggest I've seen this year in the industry. It probably is the biggest, I'm sure. And so for you to make the time to still come on and be very grounded in the way that you are, we really appreciate that. And
a good week. Not a great year, but a great week.
I'm going to, very briefly, I'm going to allow Jackie to ask a final question to you, because I didn't want to, if you've been thinking about her question, so maybe Jackie, you could finish it off.
Yeah, okay, so for you three, I think this has been a question a number of people have asked. What is the most impactful, or where do you think AI is going to have the most impact on drug discovery and development in the next 1 to 2 years?
And you've got 20 seconds each to answer it.
Yeah. Tina?
I actually think it's on the talent, which is maybe not the 1 to 2 years. To me, AI, the next generation of drug discoverers is going to think about this problem in a totally different way. And that's actually what's most exciting to me about these biotech meets tech companies, is just we're going to look at the problem differently, and that's going to change things. That's the biggest impact, not in 2 years.
I think it's a great answer, Jerome.
Yeah, I think I should probably I don't want to steal Tina's answer, but yeah, I think it changing the way that everyone thinks about the problem of drug discovery is probably the place that it's going to make the most. But yeah, generating those new ideas and making us think differently is where the most impact is going to be, I think.
So you think we're at that tipping point where that's really going to happen?
I think so, yeah.
Hannah?
So I agree with Tina, but also I think data and getting more data and getting that out there, I think is also going to have a big difference, and having the computer power in order to allow that. So that's, I think, what has been really changing this space.
Well, I'd like to thank Sunil for inviting me to chair such a lovely panel, and I've really enjoyed it. And I know there were lots of questions. I'm sorry I couldn't answer all of them, but I've tried to ensure we had a balance across all the different topics.
You've done a fantastic job of doing that. So yes, thank you very much. We have our next ChaiTime in 2 weeks, and Big Brother will be chairing the next one. And he's going to be looking at recruiting some entrepreneurs that have had some good war stories to tell about their ventures. So that should be a fascinating ChaiTime there. And on that note, I would wish you all very well and have a great weekend. Thank you very much.
Bye.
Thank you.
Bye.
Thank you.
Bye.